Yay. Yay. Okay, we started. Hi, I'm Miss Sloan, and this is a review for Honors Bio, and this is our Unit 6 Central Dogma. If you're jumping in, grab this real quick, ZQFDML. Couple things I want to go over with you. Maybe, baby. All right. So remember, we spent an entire class period just reviewing, right? So I want to use this 7th period just to clear up any questions. I brought all the reviews with us here so I can go to those slides and help you with that if that's what you wanted to do. I also want to talk to you a little bit about the framework of the test. I've altered it slightly than what you're used to. Okay. The writing part is still the same. Make sure you know your group. You still can earn 25 points there. And on your individual part, you can still earn 75 points. What's different is I'm just going to give it to you in a Google form. Okay, so that's the part that's different. So just prepare your brain. That way you're not like stressing out about it, right, when you get it. And I also did fewer questions. Okay, so fewer questions and in a Google form. So I just want you to know that. Yes?
How is that supposed to work if we're all like working on it? Are we all gonna have to bring our computers and just like be able to like work
Just like you do every single other assessment I give you when I give you quizzes. Just the same. Just the same.
And there won't be any like diagram questions because
I can put diagrams in Google Forms if I want to. No, because you can't draw a diagram, but you could do that on the writing part with your team, right? Yeah. It's just like what you've always done. All I'm doing is it's like a bigger quiz. Okay. That's all that it is. Okay. So just I wanted to tell you that so that you're not shocked, and make sure that you've got your Chromebook charged and everything ahead of time. All right. Questions or concerns about that you want to talk about? Just giving it a go, see how it works, and we might like it better. Okay. Next, maybe. There we go. So on the topics to review for this chapter, on the history, structure, and replication of that, is there anything on that you expect, or what you can do if you would like is you can highlight, and if I see a bunch of highlights on something, then I'll go over that and review it with you. Okay. But don't like highlight everything because I can't reteach the whole unit, right? So just those things that you feel like you need a little bit more help with. So let me show you what I'm seeing, okay, if it will show, because I can't do everything, okay? So I can highlight a few things on there, but I can't do everything. So I'll get into here a little bit, and we'll kind of pick and choose. But what I see, what do you see as the darkest things on there? PCR, CRISPR, DNA technology, transposomes or jumping genes, maybe a little bit on the history, and we can do the song or something for replication. All right. And then remember, you have chapter reviews that are recorded already, where I went in detail over all of these things already. They're up on my YouTube channel, and I've shared them directly onto Google Classroom. And remember, your goal is to look at those items, and I want to make sure this records over, because we said whole window, did we not? Okay, I see the red. It looks like it's still going, so we should be good. Okay, we're still here. Okay, on your history, one thing I would know, we talked about Hershey and Chase. Will it go? There we go. Hershey and Chase. They basically labeled the virus. They labeled the protein part of the virus, and they labeled the nucleic acid part of the virus. And remember, base, sugar, phosphate. So that makes sense that it's the P, right, in the DNA. And then you just have to remember S is in the protein. And By doing this, they tried to identify when a bacteriophage, what stage would this be? It would be attachment, and then what is it doing right here? Penetration. They were wondering, what is it penetrating it with that it overtakes the cellular machinery? We know now it's the nucleic acid, but they didn't at the time. So they labeled the protein and the DNA differently, and they went to see what infected that cell. And it, in fact, was the nucleic acid because they found the phosphorus inside the bacteria. Remember, they attached it, and then how did they get the empty virus to like separate from the bacteria? Do you remember what they used? A blender, and then they used a centrifuge to spin it down. Okay. Another one that I would be familiar with is Chargaff. Remember, Chargaff says every species has their own number of adenine, thymine, guanine, and cytosine, but what's similar about them is however many adenines they have the same number of thymines. Good. Okay. And then, okay, there we go. All right. And then we talked about, sorry, Rosalind Franklin. Let me see, try to get it. Oh, no. There. I see it. Let's see if I can get it to go there. Okay. How she would shine x-rays through the DNA. And when she did that, she could tell that it was helical in nature and repeating patterns and uniform in size. And then that was used by who actually came up with the structure of DNA? Watson and Crick. And they know it to be a double helix, right? And it's like a twisted, bye, sweetheart, a twisted ladder. And we talked about that the sides of the ladder are alternating what? Phosphate, sugar, phosphate, sugar. And then you can see, let me go right. Let me go right. Hang on 1 second. So you can review your structure here. When we look at the DNA, the edges of the DNA is we have those alternating phosphate and sugars, but the sugars are the ones that are connected to the nitrogenous bases. That's the rungs of the ladder. Okay. Any questions on that? Yes.
So Rosalind Franklin said on the notes that she discovered there was like a double helix, but she didn't know that it was like the sides were like that. Like
yeah, she didn't know the arrangement of the parts, but she knew that it was helical. Okay. Yeah. And she gave that information, and there's the whole like how they knew that, right? But how they got that information. Okay, next then we want to talk about how DNA replicates, and the key thing you need to know on that is that it is semi-conservative. And each old strand serves as a template for the new strand, right? And let's just go through our enzymes. So the enzymes we have to unwind and unzip, helicase, we have a leading and a lagging strand. Primers get put down one time on the leading, multiple times on the lagging. Remember, the lagging is going the wrong direction for the enzyme to read. Remember that part? So we use DNA, what? Polymerase, and it's going to bring in complementary nucleotides. And we went boom, 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 right? So if there's an A, it brings in a T. If there's a G, it brings in a C, right? And it brings in its complementary nucleotides. On the leading strand, it is continuous. On the lagging strand, it has to keep backstitching and turning around and going to make sure, because it can only move in one direction. It reads 3 to 5, making 5 to 3. What is that talking about? That's talking about those sugars, right? Deoxyribose in the DNA. Remember how we could number the sugars as we went around, right? So the key thing there, I'm not going to give you a DNA model and have you like number it, but it can read 3 to 5. It can only make in one direction because DNA polymerase is an enzyme, and enzymes are very, very what? Specific. They can only work, on their substrate in one direction, in one way. Building that complementary DNA. So you have it continuously on the leading, discontinuously on the lagging. Then you have to remove the primer and replace it with DNA, and then you seal it with ligase. Then those will twist up and become what? Sister chromatids held together by a centromere that are ultimately going to separate during anaphase. Remember, the S stage of the cell cycle of the interphase, you copy all the DNA. So if you have a book, you make another whole book. When we do transcription, that's where we want certain information for certain cells. And so we don't copy the whole book, we just copy a page out of the book. And when we do that, we're making RNA. So we still do this with me, DNA what? Unwinds, unzips, helicase, right? And in that case, you have a leading still and a lagging, but we can put the lagging strand away. And instead, since we're making RNA, we're not going to use DNA polymerase, we're going to use RNA polymerase, and it binds to the promoter. Now, it can only bind to the promoter in the case of a prokaryotic cell as long as there's not a what next to it. It starts with an R. Repressor. Remember what the repressor likes to bind to? Operator, right? So as long as it's not spilling out, then RNA polymerase can bind to the promoter. It boom booms, but when it brings in the free nucleotides, all of its nucleotides have ribose instead of deoxyribose. And wherever you want to bring in a thymine, instead you'll only be able to bring in a uracil. Otherwise, everything's the same. Then you make that mRNA, peel it off, set it aside. Okay, go get that lagging strand, zip it up, put the DNA away. Now this mRNA has to get processed. So we're going to put on a cap and a tail, and we are cutting out the introns before it leaves the nucleus, right? And we talked about levels of control. I'm jumping to another chapter just to kind of hit it all at the same time. Are you okay with that? Is all right? When we talked about levels of control, we talked about DNA being available for transcription or not. What do we call it when it's tightly condensed? Starts with an H. Heterochromatin. When it is available for boom boom, we call it euchromatin. Okay, so that's chromatin level of structure, of control. The ability of RNA polymerase to bind to the promoter and boom boom and make mRNA, that's transcriptional control. Putting on a cap and a tail, and we cut out the introns before it leaves the nucleus, that would be post-transcriptional. What do we leave behind? If we're cutting out introns, what are we leaving behind? Exons, because they will be expressed. Good. Leaves a nucleus. That mRNA has to go out and get on a ribosome. Every 3 bases are called a codon. That's the codon chart that we would look up, right? And that is complementary to a anticodon. And the anticodon is on the tRNA. T stands for transfer. It's transferring amino acids. The tRNAs land on the ribosome. The amino acids are hooked together to form a protein. What kind of control is this? Translational control. Once we have built our protein, the primary structure, we might have to what it? Fold it. That would be called post-translational control. You okay there? Let me jump on, hang on one second. Let me jump on something else. RNA polymerase binds to the promoter. Prokaryotic cells have operons. So that's what I taught you about the operon. And that includes the promoter. Then what's right next to the promoter? Operator. And who gets on the operator? Repressor, right? RNA polymerase hits landing site is the promoter. The repressor's landing site is on the operator, right? And if the repressor is there, then RNA polymerase cannot get on the promoter. It can't boom boom, right? That's okay. These genes that it's controlling, what do we call those? Starts with an S, structural genes. And in the lac operon that we discussed, those structural genes coded for an enzyme to digest lactose. If that bacteria doesn't have any lactose in it, do you need those genes? No, that's like right now your bedroom light should be off because you're not there. But if bacteria starts to have lactose in it, that lactose will bind to the repressor. Now the repressor is like, oh, I'm going to hang on to lactose now. That frees up then RNA polymerase to bind to the promoter. Boom, 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 make your mRNA. It gets translated into enzymes that are going to digest when all the lactose is gone, the repressor rebinds. Does that sound familiar? Okay, that was another whole chapter. Okay, what was your question?
So what is topoisomerase again? Do we need to know that one?
No. Topoisomerase undoes supercoiling, but you won't need to know that.
And also, so if prokaryotic cells have Alzheimer's, eukaryotic cells don't have them then?
No, they work by a different. I didn't go into the details of that, but operons are about negative controls and repressors, and eukaryotic cells are about positive control and helpers, like Boy Scouts that help RNA polymerase to bind to the promoter. They're more about yeses, not about the nos. Right? Yes. Yes, the enhancer and activators, that whole diagram I showed you. Yes, you got it. One question I didn't mention, where did the repressor come from? Who coded for the repressor? Regulator gene. Regulators, right? Regulator gene codes for the repressor. Okay, so what I just went over there was chapter 24, parts 1 and 2, and I hit chapter 25. So the other thing that you had highlighted was part 3. And part 3 was about how we can manipulate DNA and then what we can do with that manipulation, how we can use that to our advantage. So the first thing and primary thing you need to know is when you see or hear a phrase like recombinant DNA, okay? Recombinant DNA, what do you hear in that word? Recombine. If we're going to recombine DNA, DNA is really, really tiny. So to manipulate it, everything we do has to be chemically. We need to do that. So we have chemical scissors. What are they called? Restriction enzymes. They bind at a very specific spot called a recognition sequence. It's very specific. It's like these scissors can only cut felt, and another pair of scissors can only cut yarn. You see what I mean? It's very specific, okay? And because they bind at that certain series of bases, and what they end up doing is they make these overhangs, these sticky ends, okay? And if I cut 2 different sources of DNA with the same restriction enzymes, then they'll have the same base pairs exposed. And that allows me to mix and match. And then I can seal them together using what? Ligase. Okay, I can seal them together using ligase. Okay, questions or concerns on that? Okay, so those were the 2 enzymes. Then we had some other really specific tools that we talked about. One of them was PCR. PCR is like you're at a crime scene and you have a small sample of DNA. It's a machine called a thermocycler, but the process is called the polymerase. What does that make you think of? DNA polymerase, right? Polymerase chain reaction, which it sets off DNA replication. It's like doing the S stage of interphase, but it's just replicating the parts you want to use that are unique to you and you and you and you. I'm going to replicate those parts so I can find out who this person is. Now, replicating is one thing, just having more to test with. That's what PCR does. And if you remember, we looked here and we talked about how you heat it up to denature it and you add in the primers and the bases. And we learned about the COVID test, right? And then when you do that, you're forcing replication of the sequence you want. And we talked about using TAC. It's the species of Archaea, Thermus aquaticus, because it can withstand really, really high temperatures, right? Okay, so that makes us a bunch of DNA. How we might use that? We might use it in something called gel electrophoresis. And in gel electrophoresis, we use those restriction enzymes that will do what? Cut. But since our DNA is unique, how big of fragments it gets from those cuts will be unique to us. And the sole thing that gel electrophoresis does is it sorts DNA by size. But my size fragments are going to be different from George's size fragments, right? Different from Luke's size fragments because we're unique individuals. And then I can get a fingerprint, right, for me, right? Or I could get the fingerprint of a virus to see if a virus is in me, or look at evolution, right? That's a restriction. Did I taught you restriction fragment length polymorphisms, RFLPs, right? That's your DNA fingerprint, right? That's unique to you. So now we know how to cut and splice. DNA. We know how to make more copies of DNA, and we know how to identify people by their DNA. How can we use that? Well, that is all the biotechnology. And that was when I gave you those different examples of manipulating bacteria or manipulating plants or manipulating animals. But the part that I think you were a little bit worried about is, well, first of all, let me just mention this. This part I think is easy. Like, don't miss this one. Ex vivo is manipulating the DNA outside a person. In vivo, inside that person. Okay. And then genomics. What do you hear in that word, genomics? Genes, yeah. So anything that has to do with comparing sequences of DNA and similarities between that. And then I can't, let me move myself up here. Can I move myself? There we go. All right. And then we talked about CRISPR. Now, CRISPR is known as a gene editing tool because the scissor is there. But remember how before I said you have specific scissors to cut yarn or felt or whatever it is you're going to cut, right? Because they cut at a specific series of bases. This is super cool because you can keep using your scissor, Cas9, but you have guide RNA that says, here, cut here. So when we talk about personalized medicine, I needed to guide it to your DNA to fix that. And you could do more than cut. It's an editing tool. It could turn genes on, it can turn genes off, it could fix genes. So this is basically a way, just like you can edit a Word document, you can edit the words that make up the letters of your DNA. So that's huge. Add some in, turn some off, delete some, alter them. So that's a very, very powerful tool. Okay, and then I listed off several powerful tools here, like all the different things. Okay, now that took a lot of time, okay, but that was this review, and let's see, and that review, everything on chapter 24. Okay, let's see if it, move myself down here again. It hasn't found this screen yet, I don't think yet. Okay, so now chapter 25, we've talked about this right here, right? Okay, and the part we haven't talked about is cancer. So want me to just talk a little bit about cancer? Okay, so you want to know, we talked about different things like how is a cancer cell different from regular body cells, right? We went into characteristics of them. We talked about things like where, normal cells have anchorage dependence, but cancer cells don't. Normal cells, right, they have contact inhibition. If they're crowded, they won't make any more, but cancer cells will, right? We talked about abnormal nuclei, all those kind of things. But all of it can be traced back to two key genes, right? Proto-oncogenes and tumor suppressor genes. Proto-oncogenes are built good. These are good genes, right? They're in a position of control and power. There's over 100 different proto-oncogenes. But if those proto-oncogenes go bad, because they're in a position of power, bad things happen as a result of that. What's the bad things they do? They facilitate the cell cycle. And the one I always remember is RAS, because it's like stepping on the gas. It's saying go cell cycle, go cell cycle. Do you have cancer yet? No. You're not going to crash in an intersection if your gas pedal sticks down, unless what happens? Your brakes fail. Yeah. In our body, who are our brakes? Tumor suppressor genes code for our brakes. As long as your brakes work, you won't get cancer. When you get cancer is when proto-oncogenes mutate to oncogenes and keep facilitating the cell cycle, and nothing stops them from doing that. That's when you have cancer. And if it spreads to other places in your body, it's no longer benign. It is what? Malignant, right? It's metastasized. It's spread to other areas. One thing it can do is get blood vessels to grow towards it. What's that called? Angiogenesis. That's not good, right? Because then those bad cells get all the nutrients and things that they need. All right. What are the two main causes of cancer? Well, I can what? I can get it from my environment, or I can get it from my family. I can inherit it, right? EI, environment or inheritance. My family's going to give me bad genes, or something in my environment is going to mutate my genes, and that's how you get it. And then what are ways to diagnose it? Remember we talked about, you go to the doctor, you get a mammogram, you get a pap smear, you can look at it, visualize it, right? Those are all ways. And then how do you treat it? You're going to cut it out, or you're going to try to chemically kill it, right? And then we talked about future treatments as well. All right, then the last chapter is viruses, viroids, and prions. Remember when I told you I would be very familiar with the lytic and the lysogenic cycle? I'm telling you, be very familiar with it. I'm like telling you. So be familiar with it, because I'm going to ask you about it, okay, especially in your group part. And then we can go through the stages. The way I remember the stages of the lytic cycle is AP biomister. A, P, attachment, then what? Penetration. Bio would be biosynthesis. M is maturation. R is release. And how is the lysogenic different? After attachment and penetration, it does integration, and it goes into the host chromosome. And then when the environment triggers it, it will leave and re-enter. What happens after the penetration stage? What normally happens? Biosynthesis, and it goes back in there. So make sure you know those. Make sure you know the structure of the virus. So we talked about COVID-19. What are our concerns about COVID-19? Mutations. They keep mutating, and the protein spikes enable them to bind to our receptors. When we look at a retrovirus, this is an RNA virus that can work backwards into DNA and then integrate into our host cells. How are animal viruses? I didn't put that on there. Remember, animal viruses, they get our cell to take it in, and then they coat themselves in our own cell membrane so that they can hide. And we talked about emerging diseases. We're traveling to areas we've never been before, and we're cutting down trees, forests, and in those areas, and the climate is rising. And so what organisms can live where is changing, and we're getting exposed to these different organisms that we haven't had before. Viroids are naked pieces of RNA. Prions are proteins. And then structure of the virus, what do they have to have? Protein capsid and a nucleic acid core. That nucleic acid core could either be DNA, or it could be or single. You got it. And they could have enzymes and a membrane. Okay? Got it? Okay. What are you going to be taking your test with? Chromebook. Okay, so don't forget to bring that in charged. Okay, bring that in charged. What? Yeah, that's what I said. Group part is exactly the same. All right, and that's it.